The nervous system is divided into two. One, the central nervous system containing the brain and spinal cord and serves as the control center. Two, the peripheral nervous system contains spinal and cranial nerves. These serve as the afferent and efferent pathways. Let's look further at the peripheral nervous system. Sensory versus motor. Sensory neurons detect a stimulus and sends that information as an electrical signal on sensory or afferent pathways to the central nervous system. Motor pathways are motor neurons taking information away from the central nervous system on efferent pathways. There are two types of motor systems depending on the effector and differences in the motor neuron anatomy. If the effector is a skeletal muscle, that is classified as somatic. The autonomic motor system innervates smooth and cardiac muscle. These systems are controlled by different brain structures as referenced by the voluntary and involuntary notations here. Within the autonomic motor system, there are two divisions. The sympathetic division is referred to as the fight, flight, fright system. It is activated by exercise, excitement, embarrassment, and erection, which explains the E activities reference here. Everyone has had the experience of being scared. When we are scared, the sympathetic division speeds up heart and respiratory rates, changes blood flow, and inhibits the digestive system as well as many other things. The parasympathetic division is referred to as housekeeping. It is activated by digestion, diuresis, which is urination, and defecation. This division has the opposite effects, slowing down heart and respiratory rates, changing blood flow, and stimulating the digestive system, as well as many other things. Here we see a basic overview of the central and peripheral nervous systems. Let's take a look at the many cells in these tissues. There are two cell groups. Neurons are electrically active and respond easily to stimulation, so they're excitable. They perform two electrical events, graded potentials, which are local, and action potentials, which travel. The neurons are highly specialized cells. As a result, they need other cells to assist in their homeostasis. The neuroglia are cells which do just that. They help the neurons stay alive. These cells maintain homeostasis of the interstitial fluid, separate neurons, assist with making new connections between neurons, and even control blood exchange. There are 4 glial cell populations in the central nervous system, but 2 in the peripheral nervous system. Here we see the CNS cells, astrocytes, microglia, ependymal, oligodendrocyte. Astrocytes lie between neurons and blood vessels. They wrap around the blood vessels to control nutrient and waste exchange and therefore control the chemical environment of the extracellular space. Astrocytes anchor and support neurons and help neurons make new connections called synapses. Synapses are needed to store information. In other words, everything you learn, every time you learn something new, you make a new connection between neurons. Microglia are white blood cells, macrophage, that are housed in the CNS. These cells phagocytize pathogens. Ependymal cells are ciliated and line fluid-filled cavities. These cells secrete the cerebrospinal fluid, control its chemical composition, and use the cilia to move the fluid through cavities and spaces. Oligodendrocytes form the myelin sheath in the CNS. Notice that the cell body of the oligodendrocyte lies between neurons, and their extensions will wrap around many different neurons at once. Myelin sheath is an insulation tissue that helps action potentials move faster across the neuron. More about that later. Here we see the two glial cells of the peripheral nervous system. Neurolemmocytes form the myelin sheath of the PNS. Satellite cells function kind of like astrocytes in that they control the chemical environment of the extracellular fluid. They are found surrounding the cell body of sensory neurons specifically. Neurolemmocytes form the myelin sheath in a different way. Instead of one cell being wrapped around many different neurons, here, one cell wraps around one neuron. In fact, the neuron itself may have hundreds of neurolemmocytes lined up along its main structure, the axon. The beauty of this anatomy in the peripheral nervous system is that PNS neurons can regrow to an extent. This axon regeneration is not possible in the CNS and is a function of neurolemmocytes. In both nervous systems, myelin sheath performs the same action. Action potentials travel on the membrane of the neuron. Wherever there is myelin sheath, the brown ovals here, the action potentials are forced to jump from spot to spot on the neuron. Imagine you running down a hallway. You would get to the end much faster than if you tiptoed slowly. So action potentials travel fast on myelinated axons. Myelinated tissue is lighter in color, and so we refer to it as white matter. The white matter serves as highways where information as an action potential travels as fast as possible between 2 points. Non-myelinated neurons and neuron cell bodies, or soma, are found in gray matter tissue. In the gray matter, information as an action potential is saved in synapses. To summarize, white matter moves information as fast as possible, but gray matter allows you to think, feel, problem solve, and learn because the lack of the white matter enables the neurons to make new connections or synapses. The glial cells in the laboratory, you'll need to identify them simply as neuroglia, but do not have to identify the individual populations. Let's focus now on the neurons. These highly specialized cells are very long-lived, amitotic, and have an extremely high metabolic rate. They not only receive, generate, and send electrical signals, but they also communicate with each other. The structures are the soma or cell body, contains the nucleus and serves as the control center for the cell. Dendrites, receptive structures that receive stimuli. The axon transmissive structures that send action potentials. The axon hillock, where the axon leaves the soma. A node of Ranvier, bits of exposed neuron between Neurolemmocytes. This is where the action potential can jump. Telodendria, the branched ends of the axon. And synaptic bulbs, but it has many names, the physical end of each branch that will participate in a chemical signal. The soma, or cell body, contains both the nucleus and RNA-concentrated areas within called nucleoli. The RER is highly specialized and called Nissl bodies, but still makes proteins. The axon hillock is not only where the axon leaves the soma, but it is where an action potential is started. Dendrites are usually branched and tapered. They generate graded potentials, which stay on the dendrites and do not travel. Dendrites respond to stimuli and are receptive, or receive electrical signals. Axons rarely branch outside of the sympathetic nervous system. They are transmissive, propagating and sending an action potential. When many axons are bundled together in the CNS to form a macroscopic structure, we call it a tract. However, when they are bundled together to form a macroscopic structure in the PNS, we call it a nerve. A diagram and photomicrograph. Again, dendrites generate graded potentials and are receptive. The soma makes all cell products. The axon generates and sends an action potential. The synaptic bulbs, also called synaptic knobs or axon terminals, release a chemical neurotransmitter. Neurons can be classified by function. You are familiar with sensory and motor. Interneurons are the between neurons that your entire central nervous system is made of, while sensory and motor neurons are found in the peripheral nervous system. Interestingly, all neurons do not look the same. There are 3 structural classifications based on shape. Bipolar neurons have exactly 2 extensions on the soma. These are found only in the nose, eye, and ear. Multipolar neurons have more than 2 extensions off the soma. Interneurons and motor neurons fall in this category. Unipolar neurons have only 1 extension off the soma. Sensory neurons fall in this category. These do not have an obvious axon versus dendrite. With unipolar neurons, we therefore use the terms peripheral process and central process for that functional axon. The peripheral process is part of the PNS, while the central process will connect to the CNS. Now that you have met the neurons, we can talk about how neurons conduct graded and action potentials. This topic also includes the chemical events in a synapse between neurons. As mentioned, there are 2 events. Graded potentials are local changes in ions across a small spot on the membrane of the soma or dendrite. They are short-lived, dissipate, and are of variable strength. The key here is that if you get enough graded potentials, you might get an action potential started. Action potentials are longer-lived and travel from axon hillock to synaptic bulbs. They are all or none in that they happen or they don't. You cannot get part of an action potential. They are the same size every time and move in one direction only. Action potentials are only generated when enough graded potentials occur. What's electricity? Well, it is a difference in charge between 2 points. For a cell to generate an electrical signal, it must also create a difference in charge between 2 points. How? A cell uses ions, which are charged atoms. If a cell can move the ions, then it will be moving charges. If it moves the charges and creates a charge gradient, it will generate an electrical potential. The key to neurons is that they use ions to make charge gradients. Our vocabulary. Let's get this out of the way now. Some of these words will be used later. Voltage. This is a number. It is a measurement of the size of the charge gradient. Potential difference. This is the charge gradient. Resistance. This describes the relative ease at which charges can move. Insulator. Anything that makes it harder for charges to move. Here it's the myelin sheath. Conductor. Anything that makes it easier for charges to move. Here it is the neuron membrane. Again, a neuron will have to create charge gradients. Well, how will it accomplish that task? By using active transport pumps to force ions across the membrane and by controlling how and when those ions move. Sodium-potassium ATPase is the name of the protein pump in the neuron membrane. These pumps are all over the neuron and move exactly 3 sodium ions out of the cell and then 2 potassium ions inside the cell. This ATP-driven pump never stops working. In a few turns, we will see more sodium outside the neuron, but more potassium inside the neuron. Notice how the numbers are not balanced. The pump moves 3 positive ions outside, but only 2 positive ions back inside. So not only do we have a chemical gradient for each ion, but we also have a charge gradient now. It's relatively positive outside, thanks to that extra sodium ion, but relatively negative inside. This is the charge gradient needed to create a potential difference. The voltage will be the measurement of this potential difference. Now, ion channels. There are several proteins in the membrane that control how the ions cross back and forth. Non-gated, also called leakage channels. They're always open and allow the ions to diffuse across the membrane. Even though they'll diffuse because of the gradient here, the ions will immediately be pumped out again. This constant movement of ions across the membrane means the membrane responds to stimuli easily. Voltage gated. These only open and close with a specific voltage. Remember, voltage is a measurement of the size of our ion gradient. So the ion flow will control these gates. These gates are found only on axon, hillock, and axon and are responsible for the action potential. Ligand-gated, also called chemically gated channels. These open when a very specific molecule binds to them first. Some chemical receptors do this, but these are also found on dendrites and soma. They are involved in graded potentials. Mechanically gated. These channels open when they are stretched or pulled. We see these in stretch receptors. Now, both events have three basic phases to them. There are lots of details coming, so do commit this general pattern to mind. First, a neuron must create a gradient by moving the ions. Then, it uses the gradient and allows the ions to flow. Lastly, it must recreate the gradient. The graded potential is depicted here. We will discuss these again later, but for now, a ligand-gated channel binds its chemical and opens. One of the ions will flow across the membrane, but then the gates close and the pump restores the gradient. These are fairly weak and will spread a short distance in both directions from the point of the stimulus. Alternatively, another stimulus like a stretch or another chemical can also open a gate. Again, ions flow across for a little bit, but then the gates close and our pump returns the ions. Now let's look at action potentials. First, we must make the gradient, right? On the axon hillock, our pump moves the 3 sodiums out and 2 potassium in. Also, our voltage-gated channels here are closed. Can sodium cross the membrane? Yes, but it'll be pumped back in. Can potassium also leak? Yes, but it gets pumped back in. If we measure our gradients here, the voltage will be negative 70 millivolts. This magic number is called resting membrane potential. Our pump will maintain this gradient in voltage. However, if graded potentials start to occur on the soma and dendrites, the permeability of sodium across this membrane changes. More and more sodium starts to leak into the cell. More sodium means more positive will enter the cell, and that number will change. As extra sodium enters the cell, our potential difference changes. Since we are adding positive to the cell, the voltage will become more positive. If enough sodium leaks across the membrane so that our voltage rises to minus 55 millivolts or threshold potential, the sodium voltage-gated channels open. There are tons of these, and sodium will now enter the cell in very large amounts. As we are adding tons of positive sodium into the cell, our membrane voltage changes again and rises to plus 30 millivolts. This is an 80 millivolt change, a massive change for a cell. At plus 30 millivolts, the sodium voltage-gated channels will close, stopping the big sodium flow. However, at plus 30 millivolts, the potassium voltage-gated channels open. Potassium now will flow out of the cell in large amounts. Since this means positive charge is leaving the cell, the voltage drops down again. So much potassium leaves that the voltage drops past 0, past minus 55, and past minus 70. At around minus 90 millivolts, the last potassium voltage-gated channel will close. To recap, we first made the gradient by forcing ions into position. We then used the gradient by opening sodium and potassium channels in turn and letting the ions move. Now we must return the gradient. Once the last potassium channel closes, the pump can force the ions back into position and re-establish the gradient. This pump never stopped working, but it could not keep up with the massive ion flow. We can even use a chart of millivoltage to track the events of the action potential. First, in the resting state, all voltage-gated channels are closed and the pump has created our gradients. Because the membrane has a charge of minus 70 millivolts across it, we say the membrane is polarized. Now, graded potentials begin to happen and cause more sodium to leak across the membrane. If enough graded potentials occur to make the axon hillock reach minus 55 millivolts, we reach threshold potential. This change from minus 70 to minus 55 is described as depolarization. Once threshold is reached, we continue to depolarize to plus 30 millivolts. At this peak potential, the sodium gates close, then the potassium gates open. As potassium ions leave and the voltage drops down again, we say the membrane is repolarizing. When the membrane potential falls below minus 70, we would say it is hyperpolarized. Eventually, at minus 90 millivolts, the last potassium gates close and our pump gets us back to resting potential. The following terms apply to action potentials: all or none. Recall that action potentials only occur if you reach threshold potential. Without threshold potential, no voltage-gated channels will open. This means that action potentials occur in their entirety or do not occur at all. Threshold, the key to starting an action potential, negative 55 millivolts. Saltatory conduction. When the axon is myelinated, these action potentials hop on the nodes. Absolute refractory period, the period of time when the membrane cannot generate another action potential. And relative refractory period, the period of time when the neuron can only generate another action potential with a significant stimulus. Absolute refractory periods occur from threshold to just past peak. During this time, sodium-gated channels were just opened, and most of the sodium is on the wrong side of the membrane. It is impossible to reach threshold right now because gates are open and there's just not enough sodium in place. The benefit here is that this prevents action potentials from moving backwards. When one node is reaching threshold, The previous node is in the absolute refractory period. This forces action potentials to constantly move forward. Relative refractory periods occur later when the membrane is repolarizing. At this time, all the sodium gates are closed, and there's enough sodium back in place to open the gates again at -55 millivolts. This means that a second action potential would occur before the end of the first one. It does require a significant stimulus to accomplish this. Take a few minutes and see if you can put these events in order. Not every event is listed here, and that's on purpose, and will force you to think through this process step by step. Place your answers in the A&P Cafe discussion board. Action potentials require membranes, as they use membrane-bound pumps and gates. This means that an action potential cannot occur in the tiny space between neurons. For neurons to talk to each other, they have to use another method. Neuron-to-neuron communication occurs at a synapse. There are 3 synapse types: axosomatic, an axon terminal of one neuron to the soma of the second neuron; axoaxonic, an axon terminal of one neuron to the axon hillock of the second neuron; axodendritic, the axon terminal of one neuron to the dendrite of the second neuron. At a synapse between 2 neurons, the sequence will be action potential, chemical, graded potential. Step 1. The action potential will travel down the axon through telodendria to the synaptic knob. Step 2. Here, the action potential will open calcium voltage-gated channels, and calcium floods into the synaptic bulb. This is the only place on a neuron where calcium voltage-gated channels can be found. Step 3. The influx of calcium triggers exocytosis of vesicles that contain chemicals called neurotransmitters. Step 4. The neurotransmitter diffuses across the synaptic cleft space from the presynaptic membrane over to the postsynaptic membrane. Step 5. The neurotransmitter will bind its receptor, which is a ligand-gated channel. Step 6. If the channel opens and sodium moves in, the second neuron will depolarize and get closer to threshold. If the channel opens and potassium moves out, the second neuron will hyperpolarize and get further away from threshold. If the channel opens and a negatively charged chloride ion enters the neuron, the second neuron will hyperpolarize and get further away from threshold. These changes in the polarity of the membrane with the receptor is a graded potential. Excitatory postsynaptic potentials occur when sodium enters the second neuron and it depolarizes closer to threshold. In other words, it is stimulated and an action potential is more likely to occur. An inhibitory postsynaptic potential occurs when potassium leaves or chloride enters the second neuron and it hyperpolarizes away from threshold. In other words, it is inhibited and an action potential is less likely to occur. Regarding graded potentials, remember that they may not travel, but they do control the polarity of the axon hillock in a way. If enough graded potentials occur that allow sodium into the cell, the axon hillock might depolarize to threshold. This concept of having enough graded potentials occur to stimulate an action potential is known as summation. There are two ways to achieve summation. Temporal summation, when enough graded potentials occur over a period of time. Spatial summation, when enough graded potentials occur at once, and therefore there are more of them. Now, as long as neurotransmitter resides in that synaptic cleft or space, it will affect the postsynaptic membrane. For homeostasis purposes, we can predict then that neurotransmitter must be removed from the cleft to return the second neuron to rest. Here we see three options for removing neurotransmitter from the cleft. Reabsorption. The presynaptic membrane can pump neurotransmitter from the cleft back into the bulb and recycled. Astrocytes can also pump neurotransmitter from the cleft. Enzymes. The postsynaptic membrane can release enzymes into the cleft to destroy the neurotransmitter. The enzyme noted here, acetylcholinesterase, breaks down acetylcholine. And diffusion. The neurotransmitter can also diffuse out of the cleft to the extracellular space. Okay, take a moment to think through this question and discuss in the A&P Cafe. There are many neurotransmitters, and you guessed it, they can be classified in different ways. Functional classification focuses on the type of graded potential created. Chemical classifications are just that, chemical groupings. All neurotransmitters allow one neuron to control another neuron or effector. No notes needed here. This is a brief list of neurotransmitters and their chemical composition. Some are proteins, others are small molecules or gases. Definitely take notes here. The functional classifications describe the type of control. Excitatory neurotransmitters bring the postsynaptic membrane closer to threshold so an action potential is more likely to occur. This is an EPSP, and we see them on skeletal muscle cells in response to acetylcholine. Inhibitory neurotransmitters bring the postsynaptic membrane further away from threshold, so an action potential is less likely to occur. This is an IPSP, and we see them on cardiac muscle cells in response to acetylcholine. Other neurotransmitters can be both, as their action depends on the receptor present. Okay, use the free internet to quickly look up the action of Prozac as a serotonin reuptake inhibitor, and in the A&P Cafe, explain why this inhibition increases serotonin levels in a synapse. You can click on these links in the PowerPoint file to access some Quizlet flashcards.